Hello, everyone, and welcome to our first Auralis webinar, Beyond Analysis, Turning Sequencing Data into Biological Insights. I'm Giuseppina Sannino, and I am CEO and founder of Auralis Biotech, and today I am here with my colleagues, Marica Gaspari and Valerio Embrione. As I said, this is a special webinar because it's our first as company, and we are really pleased to share this moment with you. Today's agenda will include a short introduction to Auralis Biotech, and then we will explain you why biological interpretation matters and give you an idea how to go from raw data to discovery, perform a functional analysis of your data, and craft your data into narrative. Then we will end the webinar with the conclusion and summary and also a Q&A. Please feel free to submit your question during the webinar in the Q&A chat, and we will reply at the end of the webinar. So as I mentioned, I will give you now a short introduction of our company. Auralis Biotech is a young company founded in May 2025 and is based in Italy. It was founded with the idea to make your life simpler. So we want to really position us as one-stop acceleration hub that focus on solving hidden bottlenecks that holds your innovation back. And we are doing this by operating through three different divisions: distribution and on-demand sourcing, consultancy, and bioinformatics. Today, of course, we are going to focus on the bioinformatics part. And therefore, before we start, let's say, with the most interesting part for you, I would like to ask you to shortly reply to our poll. What is your bioinformatics experience? So that we can make sure that the level of talk is understandable for everyone. And please remember to close the poll after you have answered, because we'll not close alone. I still give you a few seconds. Perfect. Thank you, everyone. So now, as I said, I can leave you with the most interesting part, and I hand over the word to my colleague, Marica. Thank you.
Hello, everyone. Thank you, Josie, for the kind introduction. So today, guys, I'm going to talk to you why biology is important at each step of our analysis. So we're going a bit beyond what is the statistical jargon, and we are talking about real biology. And I titled this slide "The Illusion of Good Data" because as bioinformatician, most of our time is spent by producing clean, structured data, normalizing data, good visualization, choosing the best model for our analysis, and also automating a standardizing pipeline. But when we put our report into a context, we actually find out that we trusted blindly some fixed thresholds, or we might be ignoring batch effects, other confounders in our analysis, or we might be missing low abundance genes that are key for our analysis. So it's very important to always validate at each step against the biology that we already know and against also what we want to answer, so the research question that we have in mind. This is because statistics tell you that something has changed, but it's the biology that puts everything together. And perfect pipelines cannot really compensate for a weak biological design, because we know that biology is complex. We are looking most of the times at a screenshot of biological mechanism, so we don't see the time progression of our cells of a mechanism. And our data is very messy and it's very noisy. So that's why for true insights, we need to start with a good and robust experimental design. But instead of talking about robust experimental design, I think the foundation for it is a robust biological system. So before starting an experiment, it's always a good practice to ask these questions like, what is the best model that actually reflects my biological variability, a model that it's complex enough in order to help me to test my hypothesis, and also a reproducible model that you can also use over time, and also other people can also simulate it if you publish something. And also what is good practice effect size that we want to see, is good to carry out a good power analysis in order to check what is the estimated sample size in order to have more powerful experiments. Next slide, please. Thank you. So for a solid experimental design, it's good to start with biology. So defining a clear hypothesis, so why gene X has an effect on gene Y, and also why what is the effect of this interaction, is good to start from. And also you have some guidelines and a good framework that you can always get support from, which is called DOE, so Design of Experiments. And this helps you to actually know how to use your controls, how many controls you need, how many replicates, how to check validate your pathway competence, and also how to avoid systematic biases, or also fonts of variations into your equation that you don't need to include, the so-called confounders. So that's why here it's very important to understand also how to randomize and block your batches in order to distribute groups and avoid these biases in your analysis. So in order to make insightful discoveries, I want to talk to you about how much is important at each step, from raw data to the downstream processing of the data. It's important to do a good QC. And the example I want to take today is the RNA sequencing analysis journey. So what is RNA-seq? RNA sequencing, as the name says, is the amplification and sequencing of the RNA of millions of cells in your sample, and that provides a comprehensive snapshot of what is the transcriptome of your samples at that specific in time you consider. You probably know also the protocol for the RNA sequencing, but just to go over briefly with you, a good analysis starts on the bench. So as soon as we do library prep on the bench, we need to do our proper QC to check contaminations, library sizes. And again, after we put also our library in the sequencing platform that we choose for our experiment, we need to make sure that also here we monitor each step of the reactions that are happening. For the Illumina machines, the first thing that happens is that your library needs to hybridize to a flow cell, so a piece of glass, basically. And then there are some cycles for bridge amplification step, where you actually see this DNA that is growing and being amplified and forms clusters on the flow cell. After this, you have the sequencing itself, more cycles where your fluorescently labeled nucleotides are added to the clusters. But all this information then comes in the form of imaging data. And so we need to convert this imaging data in something that we can, in a format that we can analyze easily with our programming. So before talking in details about RNA-seq, I want to give you an overview of what are the 4 critical steps, in my point of view, in every discovery process. So the first step is the library preparation on the bench and the sequencing itself on the bench. So this step is very key for collecting the quality metrics and also the metadata associated with your experiments, like time points, treatments, controls. It's good to be very careful how we fill up this information as well. And after we put our library in the sequencing machine, we then can go on to the next step, where we convert this raw data coming from the sequencing into measurable biological information. And in RNA-seq is where we map our transcripts and we start quantifying the expression of our genes. After this step, we need to identify what is the real signal in this data. So we need to basically identify the signal to noise, and in RNA-seq is what we do at the differential expression step. And the last step, but one of the most important, is the biological interpretation of all these findings. So we translate what we know into networks, gene pathways, and then it's where we start to make sense of the biology of our experiments. As I said, in RNA-seq, but in every workflow, the quality control, it's basically important at each step, from the library preparation on the sequencing machine, and also after the sequencing itself, when we, as bioinformatician, we do QC upstream and downstream. And I wanted to give you a few examples. The first one is the image in the middle, where you can check this already on the sequencing machine. And we are looking at the clustering. So what we want to achieve is the optimal spot in the middle of the image here, where your library is well balanced. Basically, your imaging data are well detect a good clustering, because you don't want to analyze an under-clustered or over-clustered library. And another metric that is very important at each step of the sequencing is the Q30. That tells you the probability of error that one base is added to the sequencing wrongly. So the higher is this metric, and the better is your sequencing. As well, I wanted to give you an example with this image at the right of the slide about the adapters. So this is part of a report that we provide you with from the MultiQC, it's called. And you look at the adapters that are still present in your library at the top, and then after the trimming, you can see that there are no adapters left, and the QC is as we expect, and we can go ahead in our analysis. So when I talk about raw data, we are talking about FASTQ files. FASTQ files is a very complex format, but it's got a lot of information about your sequencing, about the indexes that you used, about the quality of the run that you are analyzing. So we can extract all this information and put it all together by using some publicly available tools called FastQC and MultiQC that can aggregate all your metrics into a report. And I give you some example here in this image that's at the top, you see a per base sequence biased composition library, and at the bottom you have a non-biased library that you want to achieve for your analysis. There are other metrics that you can collect, like GC content that tells you about the organism that you're studying or the coding regions that you are analyzing, and also duplication levels that may arise during a biased PCR. Or also you can check the representation, obviously, of your sequences because you know what to expect already. So after this early on QC, after the QC of the raw FASTQ files, there are other steps that are important. So the first step is the alignment of your raw data to a reference genome. And this is where we have all the annotated regions of our library. And after this step, we can check the depth, the coverage of our library, and also we can check the mapping rate, the size distribution, all things that are key to pass on to the next step, where we need to quantify these genes that we found annotated in our library. And we have what it's called the count matrix, where we have the counts for each gene, for each sample. And here what we can do is to check the counts distributions, if there are outliers, if there are a lot of zero in our counts, and so on. After this step, we need to normalize these raw counts because we want to avoid any technical artifacts or any information related to the depth or the RNA composition of our library. So we use different tools, and there are pros and cons in each of them. And what we do normally as a QC is an exploratory analysis where we do the clustering of the samples, the genes clustering, and we can early on detect batch effects, swapping of samples, so things that may save our experiments downstream. And last step, I mean, not last but least, step in our analysis is the one that everyone gets excited from, because it's called the differential expression. And this is where we convert all these numbers into a biological meaning. So after we have our normalized counts, we basically have some tools that allow us to convert all these information into the change of expression of our genes. So each tool uses different statistical modules, and it models our counts in a way that at the end we have some metrics like false discovery rate and fold change that allow us to set up some thresholds that we want to take ahead for significance and the effect size. But when we look at our Vulcano plot, we actually see a lot of genes that are spreading also over this threshold that we set. So even in this case, the biological context matters, because most of the times there is the false dichotomy between what is significant or not, because you may wonder that you may find out actually that a gene that is not that significant but has a good fold change in your analysis may be important to actually take ahead, because you need to bear in mind as well that into the validation in vitro phase, if something has too little fold change, cannot be tested, so it's not visible in vitro. So you may want to actually analyze case by case if you want to take ahead some genes that are key for your research question and your biological question. So thresholds are just guidelines. They are not the holy Bible. So they are just to reduce noise, and they are tools for you, for your interpretation and for your biological judgment. And as well, this becomes even more important when you are doing the pathway enrichment analysis or the functional analysis in order to validate your gene interactions and the patterns you see in your data. But I'm going to leave this conversation to my colleague Valerio, that is going to walk you through this functional analysis. Thank you very much for listening.
Yeah, thank you very much, Marika. And yeah, now that we reached to the point where we have the list of genes that might be either upregulated and downregulated, now what to do with these genes, besides doing a Vulcano plot or whatever other analysis upstream. The best thing to do, of course, is performing a functional analysis. Functional analysis will transform your genes, your list of genes, to something that it is biological meaningful. For doing that, you use tools like gene ontology analysis, pathway analysis, or enrichment tools. The gene ontology analysis will extract from your short list of genes after being filtered by fold change and false discovery rates into the meaningful biological processes that might be related to them. And then pathway analysis will put these genes on the map of the biological pathways, and both together will make the enrichment, will converge into enrichment tools like the overrepresentation analysis or the gene set enrichment analysis, which we'll see in a few seconds more in detail. But the most important thing is that you have to contextualize your findings by, as I said, combining the results from the gene ontology and the pathway analysis, and make them visualizing them well, first of all, in an appealing way, but also in a biological meaningful way. One very important step that you have to consider is that pathway databases are not all the same. Now, to avoid biases and to avoid incomplete annotations, one very important thing to keep in mind is that you might want to use multiple pathway databases And that's, of course, just when it's possible, because of course, if you are using, if you're studying a species which is like very commonly studied, like let's say human, of course, you can have access to multiple databases, even pretty strong. But eventually, if you are studying a less common species, you might have very limited database, number of databases that you can use. But as a rule of thumb, there is no such a perfect approach. There's no guidelines, no protocol, to actually perform this pathway analysis. But a good approach is to use, as I said, when possible, multiple databases, because it was demonstrated that this might avoid a series of problems that might arise to get good results. Now, as I said before, I mentioned overrepresentation analysis and gene set enrichment analysis, or ORA and GC. What are they, and what's the how we are going to use it? So the overrepresentation, I mean, they both answer to the question, what is the relationship between my genes and pathways? Okay, in two different ways. The overrepresentation analysis does it by using the pre-filtered gene list that you generated before. So using the fold change and the false discovery rate. It is very fast and very straightforward, and it's very good for strong differential expressions. But as a con, of course, you need to pre-filter your genes, so you might lose some good data, and you need to do this both for twice, once for the upregulated genes and one for downregulated genes. While the gene set enrichment analysis starts with all the genes that you found on your in your system, and it is very good to detect subtle and coordinated pathway shifts, and of course it doesn't need to use arbitrary cutoffs. And it goes in both ways on upregulated and downregulated genes, but on the other hand, it is quite heavy computationally. So this might be must be a little bit tricky, especially if you don't have like good supports, for doing that. Now, when to use one and when to use the other, if you already know which pathways are involved in your mechanism, or you want to explore just a few pathways that might be of interest in your system, so you go with the GC, because GC will make you exploring how genes behave in those pathways. On the other hand, if you don't know exactly, or you want to have a broad idea of what's going on in your system, so better going with the overrepresentation analysis first, and then you can move to the GC as well. So they are not mutually exclusive. And for last, now that we know how to go from the FASTQ file to the biological meaning, we have to put all this together and make a story out of that. So how we make this story? Stories are made, of course, when you talk about bioinformatics, are made by plots and reports. Here you can see an example of plots. These plots are representative of exactly the same data set in 4 different ways, so 4 different perspectives. So the message that they are delivering is exactly the same, but they do that attacking different angles of the problem. So the major point when you consider to do your bioinformatics analysis is that there's no such a way to say there's a good graph or a bad graph. they might be like more appealing, less appealing. That is true. But as long as the style and the graphic quality are solid, you need to find the best way to deliver your message. Because of course, like great figures, because the great figures will deliver your narrative. And for closing, when you have to deliver your message, of course, you have to tailor your message on the audience that is receiving it, and focus on your story just on what matters in a very clear way. And the most important thing is that you have to guide the flow, moving from what you want to show, how you want to show it, so the kind of plots, the kind of reports that you want to show, and therefore what will be the effect of this message. So like connecting the results, or moving to the next steps of your scientific discoveries. Now that we are at the end of this scientific part, let's say, and we covered like the major 4 points of a bioinformatics analysis. I want to ask you, what will be your biggest challenge? So please answer the poll, and right after, remember to close it. And with this, I will leave to Giusy for the closing remarks. Thank you very much.
I'll give you some seconds to answer, but yeah, meantime, thank you, Valeria and Marika. Everyone, remember to close the poll after you answer. Thank you, everyone, for answering. And now I'm just giving you a short summary of what you have been learning during this webinar. So we hope that you really understood how important is the biological interpretation in order to be able to transform your data into a discovery. And as Marika stressed a lot, quality control at every stage is really, really important to prevent downstream disasters. Therefore, biology first analysis means context-aware interpretation. In order to get great scientific stories, you really need to combine rigorous statistics with clear biological narrative. And if you need support from our ALICE team during your data discovery and analysis, we can help you with the free initial consultation, perform customized bioinformatics training, help you with data analysis services, or to craft your publication-ready figures, and even to build up your wet lab with affordable molecular tools. So now I would like, before to end, to answer to some of your questions. We're a bit over time, so I'll be able just to cover a few of them. Please, if you haven't added yet your question, add it in the chat, and we will take them offline, those that we are not able to cover. So I'm going to read now the first question, maybe this for Valerio. What is the ideal length of the fragments for RNA-seq, and how deep should be a good analysis?
Well, it really depends on what you wanna see. Usually for Illumina, the sequences go from 50 to 250 bases. On average, in our experience, we have seen most of the times going with 100 and 150. And how deep should be the sequence? Also, in this case, really depends on what you want to see. Like for a general RNA-seq analysis, I would say somewhere in between 20 to 30 million fragments counts would be ideal. If you want to see sequence variants or structure variants, you might need to go a little bit deeper, like 40 to 50 millions. So still really depends on what you want to do. As you I want to really stress this point: when in bioinformatics, you have to adapt your needs will adapt to the to what you want to see.
Thank you, Valerio. Then second question for Marika: is it better to go with single-end or paired-end sequencing?
I would say that really depends on what you want to achieve, because paired-end sequencing is always preferred. It increases your mapping confidence, identifies splice junctions, novel isoforms across transcripts, and improves your transcript amplification. So single-end can suffice if you're doing just, for example, in CRISPR screening, we were counting guide RNAs. So in that case, and also high throughput screening or also differential expression in simple organism, you may go with just pair-end sequencing. But still, I would prefer to go for pair-end sequencing in order to have a higher confidence.
Thank you. And we have time just for last question, but as I mentioned, we will make sure to reply to all the other questions offline. So maybe this one for Valerio: do you support also analysis with long read sequencing?
We do long range sequencing from PacBio and Nanopore. We do single cell RNA sequencing. Whatever starts with a FASTQ file, we can support that.
Okay, perfect. Then, unfortunately, we are already 3 minutes over. But we thank you everyone for joining this special webinar for us. We hope you enjoyed it, and we wish you a great rest of the day. Thank you. Bye.
Thank you. Bye, guys.